Hello, everyone. Welcome to today's live broadcast, N-linked glycosylation profiling, rapid separation techniques for the analysis of a development phase therapeutic glycoprotein. I am Matt Salem of SCIEX, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by SCIEX's Separations Division. For more information, we encourage you to visit sciex.com/CE. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button in the lower left of the presentation window. We'll try to answer as many questions as we can at the end of the presentation. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button in the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of the web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Laura Salmeron. Laura has more than 25 years of experience working in clinical and biotechnology fields, supporting protein therapeutics. She received her BS in biochemistry and biochemistry and microbiology and medical technologist from University Autónoma of Guadalajara, Mexico. She worked in RX Medical Labs in Monrovia, California for 5 years, thereafter owning a clinical laboratory in Hospital San Jose in Tijuana, Mexico. In 1995, she joined Bioden as the Analytical Sciences Group, then moved into the analytical development as the lead for the San Diego site, where she worked for 15 years. In 2011, Laura joined the Drug Product Group at Amgen Thousand Oaks as the process lead for EPOGEN program, then transferred to the Stability Group as the lead for all BITE molecules. In 2015, Laura joined Halozyme Therapeutics in San Diego, California, working with Halozyme Enhanced Platform Partners and leading the Potency Group as a key member of the analytical development team working with FIH, clinical, and commercial products supporting method transfers at CROs, CMOs, and regulatory submissions. I will now turn it over to Laura for her presentation. Take it away.
Good morning, and thank you all of you guys that are attending this webinar. It's a pleasure for me to be here. I wanted to thank the SCIEX group for inviting me to give this webinar, and I just wanted to share some of the work that we've been doing at Halozyme Therapeutics. So, having said that, let's go ahead and get started. So, just wanted to share this. Actually, these slides come from a poster that we recently presented at the WCBP conference in Washington, D.C. Last week. So all of this information is part of this poster, and obviously you will have access to it. Also, I had the opportunity to present this poster as well in the LACE conference in Santiago, Chile, which actually was a very interesting conference looking at Latin American work on CE. So just wanted to share that with you. Okay, now jumping into the presentation, as we all know, glycosylation is one of the most important post-translational modifications that may affect the efficacy and immunogenicity of the biotherapeutics, influencing the ADCCs and the CDC and sialylated species can influence inflammation. In this presentation, the complementarity of various liquid phase bioseparation methods are compared, including CE and some HPLC approaches. The N-Glycosylation analysis of a glycoprotein that we're going to be looking at is a Halozyme therapeutic glycoprotein, has multiple glycosylation sites, and how we use orthogonal methods to demonstrate how do we do characterization to understand the N-linked glycosylation of our complex molecule. And also it's more like a historical approach, because what I wanted to show on these slides is our legacy methods and how technology becomes available, how we want to continue to use these new methods that we get increased sensitivity and separation, and as well the ease of use is greatly improved. So us being a small company, we contract most of our work to CROs and CMOs. So we spend a lot of time in method transfer, and if we can transfer methods that are more sensitive and simple, and the ease of use is better, I mean, that's what we all are looking for. So this is kind of a little bit of the evolution of Holozyme, of where we started and where we want to be moving forward with our FIH program. And just, I'm not going to focus a lot about the last bullet in terms of the structures for our molecule, but as we all know, there's public database that we use with our mass spec data to measure based on their GU values, and I wanted to share some of the very fast information that we were able to obtain using the SCIEX kit method, and how we could compare it to our knowledge that we already have, and how this can help us in the future for our early molecules to get a fast read on what our glycosylation profile may be. So some of the technologies that I'm going to be talking about is the capillary electrophoresis and the LC methods that we currently use at Holozyme. So the three methods that I've been talking about is the 2-aminobenzoic acid binormal phase chromatography, and I'm going to go into actually the details of the method that we currently perform. And just keep in mind that this is our 2A method is a legacy method, and I mean it has been improved, and you can make it a little faster, but this is the current method that we perform. So it is very labor intensive. It could potentially be a little less labor intensive, but it's still quite a bit. And then we'll mention the rapid flow chromatography that we are currently validated in one of our CROs, and the new SCIEX Fast Glycan Labeling Analysis Kit that actually, I just want to say a quick story. I actually attended one of these webinars, and where they were talking about this kit, and I was very excited. So I was able to get my hands on one of those kits and try it on with our complex molecule, and this is kind of how this whole poster went about. So we got really excited with the data that we were able to do. So I'm really hoping that with this presentation I can inspire you guys also to see what's out there and give it a try. Okay, so I am going to jump into the 2AA method that we currently perform. We still perform because we're in the process of transferring for release, and for characterization we intend to continue to use some of our historical methods, but for release we're in the process of moving to more of a simplified method for some of our products, and for our new products as well. So I'm going to switch over to the next slide, because here pretty much it covers what I have in the previous slides and more of the detail, and it's always easier to see the pictures versus the text. So just kind of to give you a quick overview, this is our glycoprotein. We use about 175 micrograms, and then the denatured protein is treated with the PNGAs at 37 degrees, and the deglycosylation historically has been 16 to 24 hours. Then we have a cleanup step we use a cleanup cartridge, an E-cartridge, and that takes about 1 to 2 hours depending on the amount of samples that we have. And then after that, we have to dry our samples on a speed back, and that could take greater than 4 hours or sometimes even overnight. And then we continue with 2A labeling, and then after that 2A label is a 65 degree for up to 3 hours. And then we have another cleanup step where we use a S cartridge, and that can take, again, up to 1 to 2 hours. And then, again, we dry those samples greater than 4 hours overnight, and then we reconstitute and water, and we inject about 50 microliters for HPLC analysis. And then on top of that, I mean, our HPLC run is it's 110 minutes, so it's a pretty lengthy method, and as you guys can see, there's so many steps involved, so there's always possibility that, you know, something can go wrong or we have to start all over. And there's so many steps, there's multiple buffers that need to be prepared, and also in some of those cleanup steps, it's just we see high variability in the amount of excess dye that stays in there. So having the least, it is a good method, it is a validated method, but it is pretty labor intensive. And one more thing I wanted to mention, of course, is HPLC, so we do have to condition our columns, so that takes an additional amount of time as well. So here I'm showing you really what you guys want to see, what that profile looks like for our glycoprotein. So it does have a significant amount of peaks there. The separation is good. We are able to separate our sialylated species, but as you can see, we integrate things after probably around, I don't know exactly, but it's probably around the 15-minute time, and everything prior to that is considered part of the process of the sample prep. So now moving to a second method that we're currently using is the Rapid Fluor MS kit, and that's using the Hillick chromatography. Again, I'm going to skip this slide because I'm going to go into the flow diagram that I think it's easier to see. So here we use our glycoprotein, about 15 micrograms, and it's denatured at 90 degrees for 3 minutes and cooled down. And then the denatured protein is treated with N-Glycanase, with the enzyme for deglycosylation at 50 degrees for 5 minutes, and then it gets, the N-Glycans are released from the glycoproteins as glycosamines, which react with the fluorescence label. So we add our labeling reagents for 5 minutes, and then we add acetonitrile, and at this point we do have a little pellet that we centrifuge down, and then we just are very careful in collecting from the supernatant, and we inject about, I believe it's around 3 to 5 microliters for our HPLC analysis. So as you guys can see, this method is highly, it's a lot faster. The sample preparation is less than an hour, so it's a quite improvement to the current 2AA. And it's also mass spec friendly. Some of the hiccups that we find, I mean, the HPLC run is also 65 minutes, so it's kind of a long run. And as we all know, Hillick is very variable. So another thing that we've actually had to spend quite a bit of time is conditioning our columns. Usually to condition columns prior to being able to use them for our method, we have to spend a full day in conditioning a column. And we only get about 60 to 70, at this time, and this is in our hands. I'm not saying this applies for everybody, but we get about 60 to 70 injections per column. So if you have a lot of samples, this could be problematic. Okay, so and this is what our profile looks like. As you guys can see, it's for us, it's quite improved from our 2A method. We're getting significantly more peaks. For our, in order to set up our specifications, we actually grouped these glycans into multiple peaks because otherwise it gets very complicated. So as you can see, this is a very nice separation. We get our negatively charged glycans again at the right of our chromatogram. So I just realized, and I probably should have included the full profile, because since we don't do a cleanup step, we do get a lot of the excess dye at the beginning of our profile. But it is quite separated, so it doesn't impact our quantitation. But and I do believe you are able to do a wash step if you want. In the kit, it actually has the procedure to do that, but we didn't really think we needed to do it. But if you do that wash step, again, you're adding more time and more sample handling to the process. Okay. So now I'm going to move into the SCIEX kit that we currently started evaluating at Halozyme. And I'm going to switch over here. I mean, just to keep in mind, all of these are fluorescently labeled, so of course you would need to have your fluorescence detector. So here, in terms of the SCIEX, we have our glycoprotein, and I did want to mention that they use actually their magnetic beads, which are proprietary. So everything here is in solution, so there's really no spinning, no centrifugation involved. So it is, and for sample handling, it makes it very, very, very simple. So we have our glycoprotein, where we add our beads, our denature in solution, and denature at 60 degrees for 8 minutes. We cool down, and then we have our deglycosylation with our enzyme at 60 degrees for 20 minutes. And then followed by, so our glycans actually get captured by the magnetic beads. And then we add acetonitrile, and then we remove the acetonitrile, and all of this is actually pretty. We use a magnetic rack. I know we have just started to use this, so we haven't tested a lot of samples, but I do understand that there's a 96 well plate magnetic. You can also do this on a 96 well plate, and for high throughput samples, and that's kind of where we want to go for our QBD studies. When we have to handle a lot of samples, we do intend to use a 96 well platform. We haven't used that yet, but we currently use just our magnetic rack. And actually, as part of the kit, they provide you with a small magnetic, I think for an individual too, but you can buy these magnetic racks from anywhere. So you can increase the amount of samples that you intend to use. So anyway, going back to the method, you use your APTS for labeling at 60 degrees for 20 minutes, and then clean up with acetonitrile, and then you re-elute the glycans from the beads with water. And then the really nice thing is that your CE running time is only 7 minutes. So that, we can generate, if we have an hour sample prep, we can generate data very, very fast. And that is, for us, that's really invaluable. There's minimum buffer reagent preparation because everything comes in the kit. and obviously the operational variability is greatly reduced. We did use on high-resolution N-Cho capillary. I believe they have switched that to another capillary, but it should work the same way. So here, now I'm showing you the profile. As you can see now, we're getting even more peaks, which is great for characterization. That's kind of what we want to see. And I mean, as we optimize methods, that's what we want to get further separation of all our glycan species. Also, there is an internal standard. There's two internal standards that you include in your run. And this is actually really important. Of course, we do the corrected migration time for calculations. You need to have that. But in addition, they have provided us with a database that, based on your internal standards, and I'm not quite sure how it's called, it's a GU database that SCIEX will provide you, that you actually just open the database, and I'll show additional data on this, but I just wanted to point out that you need to have these internal standards to go into that database to get the ID of your glycans without doing any mass spec work, just to get the initial ID. I mean, of course, you want to confirm that that is accurate, but in a couple of hours, you can have a full idea of what your major glycans are, and even the smaller ones, because it allows you to zoom in and look at the smaller peaks. I mean, I'll show you some of our data, but we were able to count up to 80 peaks on this profile. So it could be very, very powerful when you're trying to do characterization and identifying some of your smaller peaks. Okay, so this is, I know, is a very busy table. I tried to put together comparing the three methods, and they all have great value, and at our lab we continue to use the three of them. So I'm not saying one is better than the other, but what I do want to focus in here is the sample preparation time going from 2 to 3 days, especially if we as a small company have to schedule time with a CRO or a CMO, our price is going to go significantly lower if they don't have to spend 3 days analyzing our samples. And for the RapidFluor, it's really the sample preparation time is a little less than an hour, and for the SCIEX it is about an hour. So, but the run time itself, I mean, 7 minutes is actually pretty wonderful. And in addition, there's no column conditioning. I mean, we all know we have to recondition our capillary, but that happens very, very quickly, and that's even built in within the 7 minutes, between the 7 minutes of your run. So it makes it very, very good. And we haven't done too much work with the SCIEX, so I can't really say how many injections you can do based on using one capillary, but based on our experience with the previous kit, you can do hundreds of injections without any issues. So anyway, I'm not going to go through this table. All this information is here, and it's pretty much just a compilation of the previous slides. So, but I just did want to focus on those two things that are very important for us. Okay, so here I just wanted to share a little of some of the work that we're currently doing. As you can see, obviously we've measured our identification of our glycans by mass spec using the RapidFluor, and even with the 2AA, we've done mass spec on that to get identification. So now we kind of wanted to confirm that the IDs that we're getting with a database that SCIEX has provided, that we're getting the same identifications, and the additional peaks that we're seeing, that we can enhance those and measure them and get an ID, just to make sure we're capturing all our glycans for our product. So here, just as you know, we did exoglycosidase digestion. We did increase the size of the capillary just to ensure that we can separate a lot of those small peaks for RID. But so this is just kind of a snapshot of, I mean, what would anybody would do to identify all the N-Glycans that you have. And here I just kind of wanted to touch into that GU database that SCIEX has provided. So as you can see, just out of right away, using this database, you can pull up your raw data file and you assign your internal standards, and then it transfers all the, I mean, you make sure that all the peaks are integrated and identified, and well, all the peaks are integrated, and then it actually it creates a file into, and it spits it out into Excel, and it gives you all these GU values, and also with the ID of the glycans, if that are in the database. If it's not in the database, you can add them, or you can customize this database to fit your molecule. So it is a pretty nifty tool that we found, especially for some of our new programs that we just want to know right away that we haven't had time to look at the mass spec identification. We right away just go in there and can get an idea of how many high manos or sialated species we have. So here I just kind of wanted to show you what the SCIEX kit looks like. Obviously it's 5 times faster than the HALEC, and we see that in the separation. And the sample prep is 1 hour, just like I said, 7-minute separation, and then you get that automated glycan ID. And it also, I don't know if I mentioned, it also comes with a pre-assembled cartridge, which, I mean, makes it pretty easy. If you're a CE user, you're used to building your capillaries, but as you get new analysts or you're working with CROs or CMOs, it just makes it a lot easier to have the pre-assembled cartridge. Yeah. So this is kind of what the kit looks like. So in summary, the evaluation of multiple N-link glycosylation profile methodologies by CE and LC-MS allows increased characterization of the therapeutic glycoproteins and supports manufacturing consistency. I mean, anybody in biotech has to do this. And this presentation demonstrates that the new SCIEX Fast Glycan Labeling Analysis Kit can be used as a complement for our current HPLC methodologies and the identification of the N-link glycosylated species. And the minor glycan structures can be easily detected and structurally elucidated by having the SCIEX Fast Glycan Labeling and Analysis Kit complement the RapidFluor HPLC-MS kit. And as a side-by-side comparison, it delivers excellent resolving power to separate positional and linkage isomers. And just to complete, the SCIEX Fast Glycan Labeling Analysis Kit has an increased sensitivity and decreased sample preparation complexity. These new methods allow to get greater confidence and understanding of our manufacturing consistency, demonstrating that all these orthogonal methods are complementary in the characterization of complex glycoprotein. And I just wanted to acknowledge some of the SCIEX folks that have been instrumental in training us and giving us support with this new kit. Anna, Lisa, and Mark from SCIEX, and Matt that is currently introduced me, and then from our lab, I mean, the additional people, Gia and Mike Labar, that is incredibly, he's our CSO, and he's incredibly supportive and bringing in new technology, even for our small company. And that is all I have. And I wanted also to thank the audience and for attending this short seminar, and and for SCIEX to inviting me again. So that's all I have.
Thank you very much, Laura. And as hopefully everybody can see, a quick poll question popped up, and if you could just take a couple of seconds to answer that. After seeing this presentation, do you plan to implement CE-based and glycan analysis in your lab? Yes, in 0 to 6 months, 7 to 12 months, or 12 plus months, or not at this time. We'll give everybody just a couple more seconds, I think, to answer that. All right, with that, I want to thank you, Laura, for that wonderful presentation. And before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we'll try to get to as many questions as we can. So it looks like we do have a couple of questions. We'll go ahead and get started with these, Laura. The first is, how many samples can you analyze using the SCIEX Fast Glycan Kit?
So the kit is intended to analyze up to 100 samples.
Thank you. The next question is, what is 2AA?
So 2 is aminoacidic acid. It is a labeling reagent that fluorescently that you can use for fluorescence labeling.
And the next question is, RapidGest PNJS-F was used for 2AA and RapiFluor methods. Do you know why the incubation times ranged from 16 plus hours to only 3 minutes?
I really don't know. I know the RapidGest that we use for the 2AA comes in a kit as well. So probably, I don't know if the concentrations or what is in that RapidGest is different from one kit to another. I'm not quite sure.
Okay. It looks like we might have at least one more question here. What N-Glycan standards do you use for CE or with the RapiFluor system?
So the standards, internal standards that we got came from the kit. So I am not sure what they really are because they're not really labeled with what they are. I don't know if SCIEX could maybe provide you that answer. I know in the past we were using maltoheptose, and now they call them D3 and D1 and D3, and I'm not quite sure what it is. I'm sorry, I don't know the answer.
Okay, we've got a couple more questions here. One is, how many samples do you typically run per week, and what would improve your workflow?
So it really depends. I mean, in our lab, since we contract things out, we are just working on our early FIH, and it depends what the stage of a program. We could be running up to 40 samples in 1 week at a time, or we can be running 5 samples. Depends on what the level of work that we're doing and how many programs we're doing. I know in our CROs, I mean, we constantly have manufacturing lots that they're testing for us. And also stability in our stability program. For some of our programs, we include glycans. As we ramp up with some of our QBD testings, we do end up getting hundreds, even hundreds of samples at a time. So it really varies. Sometimes we test 5 in 1 week, and sometimes we could test up to 100.
Thank you. The next question is it necessary to do triplicate analysis for each sample?
I don't believe we do triplicate analysis for each sample. I would have to look at our validated methods and what we do, but I don't believe we do triplicate analysis. I mean, we do have bracketing standards where we have up to triplicate of our bracketing standards in our methods, but for our individual samples, we do not.
And the next question is, you mentioned the lab is still doing all three techniques. What's the reason for this?
We are still continuing to use the 2AA method because that is the method that we have filed and it's currently approved. So until we have enough data and we get approval from the agency that we can move on to the next methods, we have to continue to do that for our program. So that's basically the reason why we still continue to do the 2AA.
Next is, what kind of injection method is used for this analysis, and will it affect sample composition? I'm assuming the question has to do with CE. It is a pressure injection, and I mean, since there's no it's not electrokinetic injection, it doesn't affect the composition because we're not selectively pulling in, so it's only a pressure injection by CE. The next question is this method suitable for glycolipids?
I'm sorry, I don't know the answer to that question. We don't really work with glycolipids, so I'm not sure.
The next question is, has an attempt at automating the sample prep been made?
We have not yet, but that's what we intend to do. And actually at WCBP, I did attend the SCIEX seminar, and they actually had some great examples on automating this and using a robot and doing 96 well plates. So I know it's possible, and they have done it. So, I mean, your best bet is contacting them and getting their presentation on that. We have not done that yet.
Okay. And there is one more question about can glycopeptides be analyzed with the P800 plus CE-LIF?
Yeah, I don't see why not.
Okay. At this point, I would like to thank the audience for all of your questions, and I would like to once again thank Laura Salmeron for her great presentation on N-Glycan profiling. Laura, do you have any final comments before we sign off?
No, I just wanted to thank the audience also for attending this presentation, and if there's any future questions you have, I mean, you can feel free to contact me. I'm in LinkedIn, and I'll be happy to share any of the information that I can.
Well, thank you again, Laura. And just as a reminder, today's educational webcast was sponsored by SCIEX Separations, and you can find more information on the Fast Glycan Kit by visiting sciex.com/fastglycan. That's FASTGLYCAN. And today's webcast will be available on demand through August of this year, and you will receive an email from Labroots alerting you when the webcast is available for replay. We invite you to forward that announcement to any of your colleagues who might have missed today's live event. And we'll see you next time. Goodbye.